You and your drug discovery team make tough calls every day. What to pursue, how to prioritize resources, and how to move faster without increasing risk. CAS BioFinder helps you get the data-backed answers you need to make informed, confident decisions quickly. Bring together expertly curated chemistry and biology content with purpose-built analytics, predictive models, and AI research agents. This shared evidence base helps you speed target validation, prioritize modalities, anticipate safety, and keep chemistry and biology in lockstep. In the next few minutes, you'll see how CAS BioFinder helps you answer the questions that drive drug discovery from cross-functional decisions to deep biology and pragmatic chemistry, so your team can move forward with confidence.
How can we accelerate research priorities with AI in CAS BioFinder?
Sometimes you just need a quick, well-sourced starting point. That's where the CAS Newton AI Assistant in CAS BioFinder comes in. Instead of spending hours stitching together data and looking for trends, you simply ask a complex question in plain language and get a concise summary with linked sources. From there, you can refine the question, redirect the results, or delve into the traditional CAS BioFinder interface for more in-depth analysis. CAS Newton also helps you explore new ideas fast. Notice a connection between two pathways? Ask for a summary of disease associations and follow up with questions about potential treatments or market activity. And because CAS Newton draws from CAS curated content, you're not guessing. You're starting from real-world evidence. It's a smarter way to move from curiosity to clarity without losing time.
How can we confidently select the best modality for our disease target combination?
When you're choosing between prioritizing a small molecule drug versus a biologic, you need more than opinions. You need shared cross-disciplinary evidence. CAS BioFinder helps you compare modalities against the biology that matters: target accessibility, pathway context, structural insights, and ligand selectivity, all in one place. Use an integrated blast search to confirm novelty, spot homologs, and flag off target risks early. You can quickly check whether your target is small molecule friendly by filtering ligands by drug likeness parameters and reviewing binding pockets. With the clean, standardized, and connected chemistry and biology data in CAS BioFinder, biology and chemistry teams align on trade-offs quickly to choose the modality that best fits your therapeutic goals and timelines. The right call up front saves cycles later.
How can we explore the untapped therapeutic potential of an approved drug?
Repurposing only works when you can connect the dots, label information, indications, primary targets, assays, and clinical evidence without stitching it together manually. CAS BioFinder gives you drug intelligence at a glance, then takes you deeper. Dig into known mechanisms, scan less common protein interactions, and filter disease associations to surface credible new paths. Further narrow your search by exploring data on non-approved indications, comparing cell lines, and confirming assay conditions. Then run a predictive bioactivity screen against disease-relevant target panels to estimate where an approved molecule might shine next. With CAS BioFinder, you move from hunch to hypothesis to prioritized repurposing opportunities quickly and transparently.
How can we stay aware of competitive challenges?
You can't plan in a vacuum. CAS BioFinder keeps you close to the market reality with clinical trial monitoring, plus links to patents and journal publications. Focus on the most relevant information, such as phase, status, or sponsor. Prioritize actively recruiting and ongoing trials to set direction and spot safety signals. Gather more information from source literature to understand who's doing what, where, and how far along they are. The result? Fewer surprises, less duplicated effort, and clearer rationale for portfolio choices.
How can we better understand the mechanisms of a specific disease?
Understanding disease mechanisms starts with clear context. CAS BioFinder standardizes synonyms and external IDs, so your team stays aligned and your data stays consistent. Survey approved and withdrawn therapies to learn what has worked and why, which proteins are involved and which aren't. Explore disease-related biomarkers. With filters for category, parameter, organism, specimen, and treatment, you can zero in on measurements that inform pathogenesis. Linked assay details and source material let you trace the evidence, build a concise mechanism brief, and point research toward the most promising biological levers.
How can we identify the most relevant targets for our disease of interest?
Big target lists are noisy. CAS BioFinder helps you rank likely targets with translational relevance, browse proteins associated with the disease, and apply fine-grained filters, such as organism, function, and experiment type, to tighten the field. Or come at it from another angle. Review biomarkers associated with your disease using brief table summaries to highlight gene-disease associations worth attention. Once you have a lead, delve deeper into measurement tables with assay context and source material to weigh the evidence objectively. Instead of engaging in subjective debates, you can recommend targets with confidence, supported by clear data behind your decision.
How can we learn how our target protein functions in the body?
To validate mechanisms of action, you need to connect structure to biology. CAS BioFinder brings protein summaries, interactive 3D crystal structures, and pathway visualizations together so you can better understand binding interactions and flag adverse events. With linked identifiers such as NCBI, UniProt, and PDB, you reduce friction across tools and teams. Visually inspect ligand and sugar-binding sites and waters of crystallization. Then, explore pathway models to see upstream and downstream effects. The payoff is clarity: how the protein behaves, where intervention makes sense, and which downstream processes you need to watch.
How can we confidently validate our target before passing it to the chemistry team?
Premature handoffs waste cycles. CAS BioFinder lets you check novelty, binding site quality, and safety signals before you engage the chemistry team. Inspect 3D binding interactions with hydrogen bonding, pi stacking, salts, metals, and waters. Conduct a blast search to assess novelty and identify homologs with similar modes of action. Explore associated ligands, their drug likeness, and pharmacology to judge potency and target selectivity. Then review predicted and known metabolites to catch potential toxicity early. When your recommendation lands, the chemistry team gets a target that's relevant, grounded in evidence, and ready for efficient design.
How can we design an antibody or peptide to deliver the chemistry team's drug to the target?
For large molecule delivery, you have to balance innovation with safety requirements and development timelines. You need sequence and structure context, plus visibility into what already exists. In CAS BioFinder, you can quickly explore known antibodies and peptides tied to your target. Review structures and sequences to understand binding properties, then navigate across disease, target, ligand, pharmacology, and references to inform selection or innovation. Whether you choose an existing binder or design a new one, CAS BioFinder helps you ground your decision in evidence, so your large molecule will be ready to pair with the payload.
How do we select the most effective binding site for our chosen modality?
Pocket selection sets the tone for everything downstream. CAS BioFinder helps you work from structure and data, not guesses. Interactive 3D crystal structures that you can adjust by resolution and organism help you focus on the most relevant information. Analyze known ligands for the target and related kinases, filtering for binding affinity. Overlay early drug likeness and bioactivity signals. Dig deeper into known and predicted metabolites to identify toxicity risks. When you need a deeper ranking of druggable pockets, export structural data for your in-house docking software. This way, you converge on pockets that balance affinity, selectivity, and feasibility based on reliable data.
How do we uncover molecule target pairs with strong therapeutic development potential?
Building a credible hit list relies on connecting modality, target, and disease data with assay details. CAS BioFinder gives you that view. With your target in mind, explore associated ligands by modality, drug likeness properties, function, approval status, and organism. Narrow the candidate search by digging into primary targets, research stage, and mechanism of action for existing drugs to direct your future research path. Deep pharmacology tables link to assays and sources so you can trust the measurements you're using. Exploring relevant scaffolds helps you understand binding options and generate analog ideas with full context, reaching a viable short list of candidates before entering the lab.
How do we optimize top therapeutic candidates from our ligand set?
Drug optimization is where time disappears, unless you learn quickly from the landscape. CAS BioFinder lets you examine non-approved drugs and failed analogs to understand assay conditions, mechanisms, and safety flags. Map nearby chemical space in 2D and 3D to reveal trends and outliers. Then investigate matched molecular pairs to test small substitutions and spot activity cliffs. Combined with early ADME and tox context, these visual and standardized data help you focus effort on modifications that are likely to move the needle and avoid dead-end paths.
How can we confidently assess the balance of potency and safety?
Potent isn't enough if your molecule is promiscuous or toxic. CAS BioFinder pairs predictive screening with substantiation visuals so you can weigh potency against risk. Screen known or novel ligands against preset or custom target panels. Then inspect the underlying measurements and confidence scores to understand why a prediction looks strong or weak. It only takes a second to review adverse events for similar approved drugs, or to scan pathways for upstream and downstream effects. With those signals in hand, you can prioritize candidates that are more likely to survive and flag likely failures before investing in synthesis or clinical trials. CAS BioFinder solves the biggest bottlenecks in drug discovery, helping you and your team make confident early decisions that reduce risk and accelerate progress across biology, chemistry, and safety workflows. By combining the renowned CAS content collection with powerful, purpose-built tools and the CAS Newton Agentic AI research experience, your drug discovery teams can validate targets faster, compare modalities, predict safety risks and synthetic feasibility, and explore repurposing opportunities. While many niche tools can help small, focused groups manage data and modeling, CAS BioFinder addresses upstream challenges to help the whole drug discovery team confidently decide what to pursue and why before committing resources. See for yourself what makes CAS BioFinder so unique. Let us know you're ready to start a free trial for your team today.